All right, so when the electrons are moving, there's two different pathways that they can take, which we call the cyclic or the linear electron flow. Now remember, that's our energy for photosynthesis. So light energy is captured, it's turned into high energy electrons by chlorophyll. That's what chlorophyll does. And then those electrons are going to be passed on. And there's two different things those electrons can be used to do. One is they can be used directly to make ATP using an electron transport chain, very similar to what happened in respiration. The other thing they can be used to do is to reduce carbon dioxide to sugar, and both of those things are going to happen. So for the linear electron flow, we're going to use an electron transport chain. But first, okay, so here's Photosystem Two. So remember, this is the one that produces oxygen as a byproduct. This is the one that cyanobacteria first evolved. We have light striking the chlorophylls. Electrons are now bouncing around, high energy electrons. They go to the reaction center, to the P680 chlorophylls, and they are bounced up to an electron acceptor. And in order to do that, the P680 P680 chlorophylls need a source of electrons to do that. And the source of electrons here is going to be water. The electrons in water have no stored energy. That energy, to bounce them up to a higher energy level, that's going to come from light. And so this is where the oxygen comes from. So Photosystem 2, not Photosystem 1, just Photosystem 2 can strip electrons from oxygen. So that means that in order to oxidize water, we have to have something that's a more powerful oxidizing agent than oxygen. And normally in biological systems, oxygen is the strongest oxidizing agent that's around. But in this case, these P680 chlorophylls are in fact a more powerful oxidizing agent than oxygen. So they can strip electrons, steal electrons from electronegative oxygen. And doing that, then oxygen loses its hydrogens because hydrogens go where the charge goes. And we're left with oxygen gas. So that's how we end up producing oxygen gas as a byproduct of Photosystem 2. Photosystem 2 then can pass its electrons onto an electron transport chain. And very similar to respiration, this is in the membrane of the thylakoids, and those electrons are passed along from electron carrier to electron carrier and are allowed to step down in energy, just exactly like happens in mitochondria. The same work is being done as a result of this, and that is pumping hydrogen ions across that membrane to create a gradient. So now we have the energy of high energy electrons is changing to the energy of a proton gradient. So the electrons go down in energy and a proton gradient is created. That is a form of stored energy. Remember, that's potential energy. And exactly the same as in respiration, that proton gradient is going to be used to drive ATP synthase molecule to make ATP from ADP. So that's the linear electron flow from Photosystem 2 to Photosystem 1 through an electron transport chain. So those electrons, what's drawing them down through the electron transport chain, remember in mitochondria, it's oxygen, that's the electron acceptor. In chloroplasts, it's not oxygen. Instead, it's the other photosystem. So photosystem 1 is what's drawing those electrons down the electron transport chain. And it has the ability to take light energy and bounce those electrons up to a higher energy level, high enough that they can be grabbed by our electron carrier, NADP+, which is going to grab those electrons and then move them on. So they go to Photosystem 1. Photosystem 1 does not produce oxygen as a byproduct. What it does is grab electrons from the electron transport chain using the energy of light again, which is striking the chlorophylls in that complex. These P700s that are at the center of Photosystem 1 bounce the electrons up to an even higher level. And once they're at this higher level, then NADP plus can be reduced. They have to be high enough energy for NADP plus to be reduced. So then we end up with NADPH, which is the reduced form. Now this molecule can carry electrons to the next step, to the synthesis reactions, and reduce carbon dioxide using that electron energy. So we have two things happening here. So electrons from Photosystem Two are going through the electron transport chain and making ATP. Electrons from Photosystem One are being used to reduce NADP+. So we have two forms of energy now as a result of the light reaction. So Photosystem 2 and Photosystem 1, that's where the light reactions are occurring. And energy is resulting from these reactions. ATP results and NADPH. Now, if there are more electrons available than can reduce NADP+, because there's a limited amount of NADP+ in the cell. So if there's not enough of this around, the electrons can also flow the other way. Okay, I'll show you that in a second. So here's a summary of that. So we have light striking Photosystem 2. Electrons are stripped from water, which releases oxygen. Those electrons are passed down an electron transport chain, which makes a proton gradient, which synthesizes ATP exactly as we do in respiration in the mitochondria. Those electrons are drawn down the electron transport chain by the P700 chlorophylls in Photosystem 1. It bounces electrons up to a higher energy level using energy from light, and then they are passed on to NADPH. So this is a kind of a little cartoony version showing you the energy, what's happening with the energy here. So we have photon energy being used to bounce electrons up to a higher energy level. In Photosystem 2, those electrons go down the electron transport chain. That energy, those electrons are dropping down in energy. That makes ATP. The electrons do not fall all the way back down to their lowest energy level. They make it to Photosystem 1. They are bounced up to an even higher energy level, high enough to reduce NADP+. And then now we have two forms of stored energy. So the cyclic electron flow, this is what happens when all of these high energy electrons cannot be used to reduce NADP+, because there's not enough of it. Instead, these electrons can go to the electron transport chain as well and make ATP. So Photosystem 1 all by itself can in fact generate both kinds of energy. It can produce ATP by shuttling electrons to the electron transport chain, and it can also store high energy electrons in the form of reduced NADPH that can go on to the synthesis reactions. So, and this is in fact what organisms do that don't have Photosystem 2. So remember, this evolved later. So Photosystem 1 evolved first, and there are still bacteria today that only have Photosystem 1. They don't produce oxygen because that's Photosystem 2. But they can have everything that photosynthesis has because they can produce ATP and high energy electrons for the synthesis parts of the reaction. So photosynthesis, but without the production of oxygen. And plants do this as well because those excess electrons are still going to be used to generate ATP. So this is an example of one of those kinds of bacteria. Purple sulfur bacteria have photosynthesis. They only have Photosystem 1. Photosystem 1 evolved first, and there are bacteria that only have Photosystem 1. Everything, however, cyanobacteria and all of the eukaryotic organisms that have plastids that are derived from cyanobacteria, they all have both Photosystem 1 and Photosystem 2 and produce oxygen as a byproduct. So there are some similarities between these two, as I've already been mentioning. The fact that The membranes are used, that inner membrane for both of these, that's the membrane of the ancient bacteria for mitochondria. It's the ancient aerobic bacterial membrane. And for chloroplasts, it's the ancient cyanobacteria membrane. The cytoplasm here of the mitochondria, the matrix, and here the stroma is where important reactions occur, which makes sense that that would be in the cytoplasm of the ancient bacteria. So we have a proton gradient in both that's created by an electron transport chain. What drives that electron transport chain is different in mitochondria. It's driven by oxygen as the final electron acceptor. In chloroplasts, the electron acceptor is photosystem 1. But it still drives the electrons to move through the electron transport chain. And of course, the ATP in both cases, ATP synthesis and the ATP is going to end up in the cytoplasm of the ancient bacteria. So in the matrix of the mitochondria, in the stroma of the chloroplast. So in the mitochondria, which we talked about in the last chapter, this hydrogen ion gradient is created across that inner membrane. And so the space in between the two membranes, what we call the intermembrane space, ends up becoming acidified, because if you add excess hydrogen ions, that is the definition of something being acidic. So we create an acidic region, so that's brown here. It was confusing before the endosymbiosis theory why in chloroplasts we have the same kind of a proton gradient, but the region that becomes acidified is actually inside the thylakoids. Because if this is outside the ancient bacteria, what about this? Isn't that inside? Isn't this the reverse? And in fact, this space, the thylakoid space, as we call it, that's the space inside, is actually equivalent to this space. And that becomes more clear if we look at a cyanobacteria. So here's a close-up of a cyanobacteria. We call their foldings of the membrane thylakoids as well. If you remember when we talked about prokaryotes, I said that the vast majority of prokaryotes do not have any internal membrane structures. Well, the cyanobacteria are an exception to that. They in fact do have foldings of their membrane, which we can see here. And we call those thylakoids, and they are in fact connected to the outer membrane of the cell. So instead of being disconnected like they are in chloroplasts, they are in fact continuous with the bacterial outer membrane of the cyanobacteria. So things that are inside, in between these thylakoid membranes, in a cyanobacteria, that's actually continuous with the outside of the cell. And so that's equivalently outside the cell. So the reason that the protons build up inside the thylakoids and also in the intermembrane space of mitochondria, those are both equivalently outside the cell in both cases. So the ATP and NADPH are going to end up in the cytoplasm of the ancient bacteria, which in chloroplasts we call stroma. So that's inside the cell. The thylakoid space inside the thylakoids is equivalently outside the cell, and that's more clear in chloroplasts than it is in plastids. So here is our photosystems, Photosystem Two, bouncing electrons up to a higher energy level, producing oxygen, passed down through the electron transport chain. Protons are being pumped into the thylakoid space. Those electrons hit photosystem 1 and are bounced up to an even higher energy level, and they are passed on to an electron carrier to the enzyme, which then can reduce NADP plus to NADPH. And now we have our two energy sources in the stroma, so in the cytoplasm of the ancient bacteria, being passed on to the synthesis reaction. So that's the light reactions. What happens with these two photosystems? So what goes in and what comes out at each step is important to know. And just like for respiration, drawing it out is going to really help you to remember what goes in and what goes out of each step. So draw a big chloroplast, draw a thylakoid, so you have the stroma and the thylakoid space labeled, and draw what goes in and what comes out at each stage.